Thanks for the love, Kevin and Bob and Cecile. The mic to you, Cecile. Thank you.
Welcome to Quantum Photonics. Here we are with this very interesting topic, and this is really very relevant to us. So, while newly approved drugs for Alzheimer's show some promise for slowing the memory robbing disease, the current treatments fall far short of being effective at regaining memory. What is needed are more treatment options targeted to restore memory, and so that will be the topic of our guest speaker here. And since most of the current research on potential treatments for Alzheimer's focuses on reducing the toxic proteins such as tau and amyloid beta that accumulate in the brain as the disease progresses, the team veered away from this route to explore an alternative. So, yeah, rather than trying to reduce toxic proteins in the brain, we are trying to reverse the damage caused by Alzheimer's disease to restore memory, said our guest speaker, Dr. Tara Tracy, in an interview. And the findings of this paper appear on the issue of the Journal of Clinical Investigation. So, I would like to tell you about our guest speaker. Dr. Tara Tracy has expertise in synapse biology and in research on Alzheimer's disease and related dementias. After graduating from Wesleyan University, she received her PhD in neuroscience from University of California, Berkeley, where she studied synapse development. During her postdoc training at the Gladstone Institute of Neurological Disease and the University of California in San Francisco, Dr. Tracy investigated the toxic mechanisms that drive neuron dysfunction and cognitive decline in Alzheimer's disease. Dr. Tracy joined the Bach Institute faculty in 2018, and her laboratory is investigating how synapses are altered in the brain, leading to cognitive decline in aging and in neurodegenerative diseases. The research in Dr. Tracy's laboratory has been funded by the National Institute on Aging, the Alzheimer's Association, and Forever Healthy. Dr. Tracy has received awards in recognition of her work from the Alzheimer's Association and the Society of Neuroscience. And in 2022, Dr. Tracy was awarded the McKnight Brain Research Foundation Innovator Award in Cognitive Aging and Memory Loss from the American Federation of Aging Research. So, without further ado, I proudly bring to you our guest speaker, Dr. Tara Tracy. Tara, welcome to Quantum Photonics.
Thank you so much, Cecile, for the lovely introduction and for your work and the work of the other organizers to put this together. And thank you again for the invitation. It's a pleasure to tell you all about our work from my lab today. Have provided some of the slides showing some of our work that I'll be talking from. So I'll start out by saying that the overall title of my presentation is called Repair and Resilience. And our goal is to fix dysfunctional synapses and tauopathy. And again, as Cecile said, I'm located at the Buck Institute for Research on Aging, which is in Marin County in California, and I'm an assistant professor here. And so the focus of my lab in general is investigating the mechanisms underlying the memory loss and the cognitive decline in Alzheimer's disease and other tauopathies. And I'm sure everybody's familiar with Alzheimer's disease, but I want to first explain what tauopathies mean. And so I have a slide number 3 here to explain what tauopathies are. They are a class of neurodegenerative diseases that are age related, so they occur with age. And tauopathies include Alzheimer's disease, which is the most common type of tauopathy. But there are other types of tauopathies that I have listed on the slide, including Pick's disease, CTE, PSP, and several others. And the thing that's common among these different neurodegenerative diseases is that there's accumulation of a protein called tau in the brain, in the brains of these people who have these diseases. And there's a picture showing what this kind of pathological accumulation of tau protein looks like in the brain on that slide. And so, we think there's some commonalities across these diseases, but there's also some differences in terms of mechanisms that are causing dysfunction. But in general, for the commonalities, most of us in the field think that this accumulated tau protein in the brain becomes toxic in these diseases. And when it becomes toxic, we think it causes this dysfunction or pathophysiology in the brain, which can lead to neurodegeneration. And both of these things can cause memory loss and cognitive deficits. So many of us really want to understand what are the mechanisms underlying these effects of tau. And in particular, my lab is very interested in how toxic tau causes pathophysiology. So I have a little question mark on my slide 4 there to try to tackle this question of how does toxic tau cause pathophysiology. And on the next slide 5, we think that tau has a particular effect on the synapses in the brain. And the reason we're interested in this is because synapses are critically important for memory and cognition. We have over 100 trillion synapses in the human brain, and they play a very important role in that they are required for the transmission of information from one neuron to the next. And these synapses, the activity at these synapses is what is responsible for encoding memory after a learning process. And so you can imagine what I'm going to show you on the next slide, which is slide 6, that synapses become dysfunctional in neurodegenerative disease and tauopathies in particular. And so what I have shown on slide 6 is that much of our research suggests that the dysfunction of these synapses is what happens in the earliest stages of cognitive decline in disease. In particular, it's highly correlated with the accumulation of the pathogenic tau in the brain. And so we think it's a really important process to try to understand better the mechanisms that are leading to the loss of cognition. The actual neurodegeneration or the loss of neurons tends to happen at the later stage of the disease. And that is also the hardest time to treat disease at the end stage. And so what we're really interested in doing is if we better understand the mechanism for the synapse dysfunction, we're hoping to come up with new treatment strategies to extend health span effectively and restore memory at least at the earliest stages of the disease process. And so next, I kind of just said this already actually on slide 7. Our goal is to really repair the synapses or the damage that's been done in order to rejuvenate the brain in Alzheimer's disease and related dementias. We have a model here where we think, again, toxic tau in the brain is causing synapse dysfunction, which leads to memory loss. And we think we have figured out a way that we could be able to repair synapses and restore memory. And so now I'm going to talk about this mechanism for synapse repair that we've been studying. But I first need to give a little more background on how synapses work. So on my slide 8, I have a picture of a typical glutamatergic synapse, and this type of synapse is represents the majority of the what we call excitatory synapses in the brain, and they are very, very important for encoding memories in the brain. And these synapses have the neurotransmitter called glutamate. And so we have a presynaptic terminal containing with the glutamate containing synaptic vesicles, and then we have a postsynaptic site which contains the glutamate receptors, which are the receptors that transmit the information from the neurotransmitters to the postsynaptic cell. And I want to point out a very important physiological attribute of these glutamatergic synapses is the fact that they can have a persistent change in their strength of their encoding. And we call this, in neurobiology, we call this long-term potentiation. And that's LTP for short. So LTP at these glutamatergic synapses is widely believed to be the cellular basis of learning and memory. And so this is how we think memories are formed in the brain after learning. And so here again, we have a picture of a synapse on my slide 9. We have a picture of a synapse on the left that contains the glutamate receptors. And when there's a certain type of pattern of neuronal activity which happens during a learning event, these synapses are strengthened by the addition of additional AMP receptors. So you can see in the image on slide 9, you can see there's an insertion of AMP receptors at the postsynaptic surface. And what this does is this will enhance the signal at that synapse and strengthen its response. And this is thought to be how the memories are encoded, by the strengthening of the response. Now, what we know in tauopathy on slide 10, in tauopathy, we know that there's toxic tau at synapses, and there's accumulation of toxic tau in general in the neurons. And we know that this blocks the recruitment of these glutamate receptors to the postsynaptic surface. And when that's blocked, there's no longer LTP at the synapse, and that means the synapse cannot encode the memory. And so we think that this pathophysiology, the specific loss of LTP at the synapses, is driving a lot of the memory problems, at least in the early stages, before there's the loss of neurons in the later stage of disease. And so my lab is very interested in this process, or this impairment of the LTP at the synapses. So we've been working on this a lot to try to better understand it. And we've become interested in a protein called KIBRA, which is on slide 11. And this KIBRA protein is highly expressed both in the kidney and the brain. And the KIBRA that's expressed in the brain is expressed in the postsynaptic side, and it regulates a number of things at the synapse, but importantly, it's required for LTP, and there are mice that are deficient in the KIBRA protein have impaired memory, so it's also important for memory formation. The other interesting things we know about KIBRA, this protein, is that there is a polymorphism in the gene that encodes KIBRA in humans. So there's a human polymorphism that is associated with memory performance. So people who have a certain allele in their KIBRA gene um, are better at memory tasks than others. There's some evidence for this in the literature. Also, that allele is associated with risk for late onset Alzheimer's disease. So this is some evidence that we already knew that KIBRA was linked to Alzheimer's disease and to memory performance. We also know that KIBRA acts like a scaffolding protein, which means it binds to a whole lot of other proteins at the synapse. At least 20 different ones have been identified. And so this suggests that it could play an important role in the function of the synapses. And what we have found specifically is on slide 12, we found that there's a KIBRA deficiency in the brain that we think is underlying the memory loss in Alzheimer's disease. So this is just, the slide 12 is a model showing that a healthy normal synapse has KIBRA and low levels of tau, and that with LTP, you get a strengthened response representing the formation of a memory. But in Alzheimer's disease, we have low KIBRA levels, and we think that's preventing the receptors from being incorporated at the synapse, and the synapse is not strengthened, so there's impaired memory. So we think this is an important mechanism. So the next question that we wanted to ask after finding that was, can we enhance KIBRA signaling in order to repair the LTP in the neurons in the context of toxic tau? And so that's what my lab set out to do in the paper that we recently published in JCI. And so on slide 14, I have the title of our paper, which was KIBRA Repairs Synaptic Plasticity and Promotes Resilience to Tauopathy-Related Memory Loss. And this was a study that was co-led in my lab by Dr. Grant Kawe and Dr. Christine Pareja Navarro, who did a tremendous job with the efforts for this paper. And so just briefly, I want to start out telling you about the work, very briefly about the work we did to look at KIBRA as a potential synaptic biomarker in tauopathy. So we found some really interesting findings in both human brain and in human CSF. So on slide 16 is our data showing the analyses of KIBRA and tau that we did from postmortem human brain samples, both from controls and from cases with Alzheimer's disease and Pick's disease, which is another tauopathy. And interestingly, we found that reduced KIBRA levels in human brain correlated with increased levels of pathogenic tau. And the pathogenic tau that we looked at specifically was the modified tau that's acylated and phosphorylated, which is oftentimes the tau that's associated with pathology, especially in this case. And so, so this suggested that there is a relationship between the KIBRA levels in the brain and the tau levels in the brain, which is very interesting. And further, we wanted to look at the relationship between KIBRA and cognition, or KIBRA in the brain and cognition. And so we had data on the clinical dementia rating for the cases that we had. And this is on slide 17. And the control cases that had a low clinical dementia rating, meaning they had pretty much intact cognition at time of death, had higher KIBRA levels in brain than those that had more moderate dementia, which had lower KIBRA levels in the brain. And we saw a very strong correlation between the degree of cognitive impairment versus the levels of KIBRA. So this was very interesting, we thought, to see that suggests the very strong relationship between KIBRA in the brain and cognition in people with neurodegenerative disease. Next, on slide 18, we looked at KIBRA levels in cerebral spinal fluid. And the reason this is important is because, we can't look at KIBRA in the brain of living people. We can only look at it in the brain postmortem samples. However, we received CSF from living patients, and this is important because it could serve as a biomarker. It could tell us something about maybe how much KIBRA could be in the brain. We're trying to do that for future studies, but in this case, we see also a very strong relationship between KIBRA levels in CSF and tau levels in CSF. And the interesting thing is that it's an opposite effect, where we see more KIBRA in the CSF associated with more tau in the CSF. And if you remember, in the brain, we saw less KIBRA in the brain associated with more tau. And we also saw people with the MMSE, on slide 18, there's a MMSE test of cognition that we compared to the CSF KIBRA levels. And we found that the people who were more impaired in the MMSE score also had higher KIBRA levels. So in summary, our human findings suggest that more KIBRA in the brain is good, and that's associated, we think, with less KIBRA in CSF. And on the other hand, people with more pathological tau in the brain and in CSF were associated with less KIBRA in the brain and more KIBRA in the CSF. And this was also linked to cognitive impairment. So we think that the KIBRA levels in general could be kind of a biomarker to tell us, how well synapses are functioning in the context of tauopathy. Next, I'm going to talk more about our mechanistic work in our mouse models. And so on slide 21, this is where we're really starting to see, can we restore KIBRA signaling to rescue the LTP. And so we looked at the KIBRA sequence and to try to come up with ways to, modify it. And we tested it. Actually, I'm going to skip to on slide 23. What we did was we decided to take a piece of the KIBRA protein that we thought could be, the one that could have the best effect. And this one we called CT KIBRA, and it's basically a small fragment of the C-terminal domain of the KIBRA protein. And we tested it in our tauopathy model, which I have described on slide 22. So in this paper, we used the tau-KQ model, which is a mimic of hypercetylated tau that is found in almost all of the tauopathies. So it's found in Alzheimer's disease, but also other tauopathies. So it's kind of could have a broad impact in terms of tauopathy. These mice develop memory deficits, and they also have impaired LTP at their synapses. And so we thought this was a good model to test whether KIBRA would have an effect at repairing synapses. So what we did is we looked at, I'm on slide 24, in with the tau QQ model. We find that the AMPA receptors, and this is one of the figures from the paper. I'm not going to explain it in detail, but I'll explain a little bit. We were looking at the AMPA receptors at the synapses, and the GLUA1 is a subunit of an AMPA receptor, and with LTP, when we induce LTP in the neurons, in control neurons, we see an increase in the AMPA receptors containing GLUA1 at the synapses. Tau KQ impairs that. It blocks the AMPA receptor recruitment. However, the CT Kibra, when we express CT Kibra with the tau KQ, we rescue the AMPA receptor trafficking in LTP. We did try another version, another truncated version of Kibra that was from the N-terminus, but that didn't have the same effect. So that's why we focused on this CT Kibra fragment for the rest of the paper. So we made a lentivirus to express, in slide 25, we made a lentivirus to express the CT Kibra, and we did some electrophysiological recordings where we could look at the LTP at the synapses in the mice that express the tau KQ. And these guys are aged mice, so we hope to model, the age-related diseases. And we do find that the expression of the CT Kibra in the hippocampus of these mice rescues the LTP of the synapses in these tau KQ mice. In slide 26, we ran some memory tests on the mice that were expressing the CT Kibra. We ran several tests in the paper, so maybe I won't go through them in detail, but they were all hippocampus dependent. So the hippocampus, I should say, maybe is the region of the brain that we think is most important for the formation of new memories, that is also most affected in Alzheimer's disease. So, with these mouse models, we're trying to model that in the lab, and we find that CT Kibra can improve the memory in the mouse model that we're using. So that was very exciting to see that we not only did we have a functional effect at the synapses, but we also had a cognitive effect on the mice with the CT Kibra expression. The other interesting things that we found were that on slide 27, we found that with expression of the CT Kibra in the brains of the mice, it did not affect the amount of tau that was accumulating in the mice. And so what that means is that the CT Kibra restored function without reducing any of the toxic tau that was around. And we think that's what was promoting the resilience in the brain, or the resilience of the synapse function. Moreover, we also still saw synapse loss in the brain, despite the improvements in the cognition and the improvements in synapse function. There was still some synapse loss, which means that the circuits could still function in a positive way, despite having some neurodegeneration already. So that was very encouraging. And then lastly, I'm just going to briefly mention that we, at the end of the paper, we looked into the mechanism for how does CT Kibra have this positive effect on the synapse. So mechanistically, we looked into that. And what I have on slide 30 is we found that the CT Kibra repairs the LTP by stabilizing another protein called PKM zeta. And this PKM zeta protein has been studied for many years by others, showing its role in plasticity and in synapse function. And so we basically showed that CT Kibra was binding to this PKM zeta and stabilizing it at the synapse. And we think this is a mechanism that contributed to how it was repairing the synapse function for plasticity to occur. And so I think this is a really interesting way that to promote resilience, that's quite a bit different from other ways that people are developing therapies in the field. So in the end, I think I'm going to say some future directions here, and I think maybe we can have some discussions. Uh, but the future directions we're thinking is, how can we make this a therapeutic? And so my lab is slowly working towards this. This is a long process, but we've designed a peptide, so a smaller peptide based on the work that we've done in the Kibra sequence, that we think someday could be potentially developed into a therapy that could be delivered to people, which would be amazing. The second thing that we're working towards is working out how Kibra could be a synaptic biomarker for patients. And this is important because we want to have some kind of biomarker to know who would benefit the most from having a Kibra peptide therapeutic. So we think this is really important to do together with developing a therapy. And the third thing that we're interested in is, I mentioned there's a polymorphism in the gene that encodes Kibra that's related to memory and to risk for Alzheimer's disease. And so we want to better understand that polymorphism and understand why some people may be more susceptible to getting dementia, because they have that polymorphism in the gene that encodes Kibra. So that's kind of our future directions from this project that was recently published. And then I have my last slide is just acknowledging my lab and the people who collaborated with us on the project and our funding. And we couldn't have done any of this without all of them. So I have to acknowledge that. And I think that is where I would stop.
Yeah, thank you so much, Tara. So, for those who just came in, please check. We have the slides at the top of the screen, and here on the room chat, we had posted a popular article that had featured the work of Tracy Lab. That is the lab of Tara, called Tracy Lab, and of course, the paper that she is talking about. And I would like to just have a short reintroduction of our guest speaker here for those who just came in. So, yeah, our speaker, Dr. Tara Tracy, is a PhD and an assistant professor at the Bach Institute for Research on Aging at the University of Southern California. And her expertise is on synapse biology and in research on Alzheimer's disease and related dementias. After graduating from Wesleyan University, she received her PhD in neuroscience from University of California, Berkeley, where she studied synapse development. And during her postdoc training at the Gladstone Institute of Neurological Disease and the University of California, San Francisco, Dr. Tracy investigated the toxic mechanisms that drive neuron dysfunction and cognitive decline in Alzheimer's disease. Dr. Tracy joined the Buck Institute faculty in 2018, and her laboratory is investigating how synapses are altered in the brain, leading to cognitive decline in aging and in neurodegenerative diseases. The research in Dr. Tracy's laboratory has been funded by the National Institute on Aging, the Alzheimer's Association, and Forever Healthy. Dr. Tracy has received awards in recognition of her work from the Alzheimer's Association and the Society for Neuroscience. And in 2022, Dr. Tracy was awarded the McKnight Brain Research Foundation Innovator Award in Cognitive Aging and Memory Loss from the American Federation of Aging Research. So we'd like to announce to the audience that we are opening the floor for questions. Please feel free to raise your hand if you have questions to ask. We will bring you up the stage. I would also like to acknowledge the presence of Nas and Bulkar, who came here as speakers on stage. And so I wanted to ask a question, but I think I would like to pass the mic to Nas, who came in first. If Nas would like to ask a question, we are trying to do this in PTR order. So I'll give the mic to Nas and thereafter to Bolkar. And maybe if Bob would like to read some questions later on, if there are questions or comments on the room chat, we will do that. So Nas, may I give the mic to you?
Sure. Thank you, Cecile, and thank you, Dr. Racy. Um, very well done, thoughtful uh, presentation. I was following the slides while you were speaking. So, my question, the title it said to repair damaged synapses in Alzheimer, has been any targeted therapy been identified in your research so far? And what mechanism would you use to repair a damaged synapses? You mentioned the genetic, the defective gene. Would that be gene therapy? Would that be protein therapy? What targeted therapy would you recommend that can be helpful in finding a way to repair that synapses damaged, the damaged synapses?
Oh yeah, thank you for asking. So we think that uh, our paper, we expressed the CT Kibra sequence. So it's a piece of the Kibra protein, and we think that that was sufficient to repair the dysfunction of the synapse. So it's a functional repair of the synapse. And so, however, we acknowledge that expression of the full CT Kibra by lentivirus is probably not going to be the most ideal way to make a therapy, right? Because there are not many currently, I think, getting a viral expression of something into the brain is going to be harder for an actual therapy for us. And so we're taking a different approach now for our future studies where we're making a much smaller peptide uh, that from the same from the a smaller piece, even a smaller piece of the CT Kibra sequence um, that we think could be delivered um, potentially into the brain without needing lentivirus or any kind of maybe really hard delivery method because it is very difficult to get things or drugs across the blood-brain barrier for treatments of neurodegenerative disease, and so we're very well aware of that. And so rather than trying to deliver a larger protein, we're right now we're trying to deliver a small peptide based on the KRAS sequence that we think could provide functional repair of the synapses to promote memory formation. Does that answer your question?
Yes, it does. Of course, the virus concern is extremely legitimate and um, well thought through. It's possible to try or explore CRISPR or some sort of a small peptide, as you said, because the brain is always difficult to have any drugs, brain barrier drugs that's safe. It is a challenge, but the thought is it's promising for sure.
Yes, yeah, thanks for bringing that up. Yeah.
Uh, Thank you so much, Nas, and thank you, Tara. So, if you have other questions, Nas, we will circle back to you later on. In the meantime, we'd like to give the mic to Bolkar. Bolkar, I'd like to give you the mic now.
Okay, thank you very much. So I'm going to ask the questions very fast. So whether the 2 protein creates the less Cubra deficiency or less Cubra deficiency is a part of the piling of the 2 protein, because some say it's not just limited to 2 pathologies, but it's also a part of inflammation, neurological loss, and amyloid beta accumulation. And the second question I have is that removing or reducing tau pathology, in what extent can the brain heal itself?
Those are great questions, and probably, as you know, very, very difficult ones, because you're right. There are a lot of things happening in the brain in these diseases. And you're right, inflammation. There's also, in the case of Alzheimer's disease has the amyloid pathology as well. So yeah, that's a uh, very important point. And so we have only tested our CT Kibra strategy in mice that have pathological tau. We have not yet tested it in mice, like mouse models that have amyloid beta pathology. This is something that we plan to do. And so that's going to be important to address that question. Um, uh, Inflammation, there's many studies suggesting that the tau pathology can cause inflammation. And so, it's possible that that the CT Kibra repair, the functional repair of synapses, could occur despite the inflammation that's happening in the brain due to the pathological tau. And so that I think that's definitely possible. Although again, it might depend on the stage of the disease. Meaning, probably at the end stage of disease when there's a lot of inflammation and a lot of neurodegeneration. We think that at that point, our type of therapeutic, where it's functional repair, we think it may not be as useful. So that's why it's important. I think you mentioned the second question. You mentioned about, I think it was reducing tau. Sorry, can you repeat that second question again?
Okay, so the question is like uh, the piling of the tau protein does it create less deficiency of Cubra, or could it be vice versa? And I want to add another comment on this. Do you think if we can find a way to segregate tau protein piling up in the brain, can we stop the process in the beginning, or we do not know yet?
Oh, yeah. So there are many people working on clinical trials to reduce the tau protein in the brain as a way to treat, for example, Alzheimer's disease or other tauopathies. So there are many people interested in this, reducing the toxic tau proteins. And I do think that's very important that we have that, and hopefully those therapies will work. And I also imagine that, let's say we slow down the disease, and that we slow down the cognitive decline by reducing tau levels in the brain. I would not expect that to recover function. I think, reducing tau levels will slow down the progression, but I would not expect it to repair function, for example. And so that's where our strategy, I think, could be useful in that, if you have a therapy that reduces tau in the brain plus a therapy that repairs synapse function, I think this could be the best case scenario, as a kind of combinatorial therapy to get the best outcome. That would be my my thinking on what could be best. And so I hope that answered your question.
Like, just to finalize, so in the first place, if there was no piling of tau protein, there won't be any Alzheimer or cognitive disorders. Is that that we can understand?
Oh, thank you for asking that. Yeah, so uh, there are obviously other neurodegenerative diseases that don't necessarily involve tau. Although, it's really, it is really quite striking that there are so many different diseases that do involve the accumulation of pathological tau. But yeah, is how would you say, in like normally people who age with kind of normal cognition, or there's always some cognitive decline with aging, but it's very different from developing dementia. So in those people, I would expect that probably most of them don't have the same kind of pathology. I don't think this was part of your question, but there is a really interesting point around that area where there are some people who age without getting dementia, but that do have the tau accumulation in the brain. And so these are very interesting people who have tau accumulation in the brain, but have intact cognition with age, and they don't develop Alzheimer's disease. These people would effectively be resilient to the toxic proteins in the brain. And these people are very interesting, right? But I would say that most people who are healthy would not have this accumulation of this bad tau protein.
So we can also say the Kibra deficiency could not be related to accumulation of the tau protein also.
Oh, so we do know that tau, like pathogenic tau, can cause Kibra deficiency in our mouse model. So we do have evidence for that from our mouse model. We cannot say that for humans, because we can't do mechanistic studies on humans. So all of our human data is correlative. So we cannot, in humans, we cannot establish causation. But in our mouse models, it does look like pathological tau causes Kibra deficiency.
But can you have Kibra deficiency without the accumulation of tau protein as well?
Oh, thank you for saying that again. Okay, so there is some in aging. My lab has done a little bit on Kibra in aging. So if in mice, so if we look at aged mice in the brain, we do see slightly lower levels of Kibra in aged mice compared to younger mice. We do not know if this is associated, like we haven't done enough work yet to know whether this is associated with age-related memory loss in mice. So this is something we're interested in. So there could be, in the absence of pathological tau, there could be relationships between age and Kibra deficiency and memory that we're very interested in looking at further.
Yeah, thank you so much, Volkar. Bob, were you about to speak? I'd like to pass the mic to you.
I was just going to step in case you were otherwise occupied. So back to you, Cecile.
Yeah, I'd like to pass the mic to you if that's okay for now.
Oh,
We
sure
have two
thing.
more.
In my PTR, I think Kevin would be next. Do you have a comment or question for us, sir?
Oh yeah, yeah. I was just wondering if you heard about the recent study researchers at the Sinai Cedar Sinai about the working memory. Uh, they identified that if this ties in with that, because this has to do with Alzheimer's and ADHD, attention deficit hyperactivity disorder. Um, they identified brain cells responsible for working memory, the type required to remember a phone number. I shared this yesterday at the news here. So of course I'm going to share this, ask you about this today. And it's, Sword, because you were coming on, and in Berkeley, I'm in Bay Area. God, Berkeley's my favorite uh, university as well. And so I don't know if you're aware of this study, if it ties in with the brain waves. Um, they identified a group of neurons influenced by two types of brain waves that coordinate cognitive control and the storage of sensory information and working memory. And these neurons don't contain or store information, but are crucial to the storage of short-term memories. And the findings could have implications for understanding and treating conditions that affect working memory, such as Alzheimer's and ADHD. As I said, if you've heard about this study.
Yeah, thank you for raising that. I was actually just looking at I just looked it up, and it looks like it was a study done in humans, which is very cool. Yeah, so in terms of how this could relate to our work, so we do have ways of we have a memory test that we perform on the mouse models of tauopathy, that it's a memory test for working memory. And we do see impairment in working memory in our tauopathy mice. And the the CT Kibra that we expressed did reverse the working memory deficit in the mice. We don't know the mechanism for that. We don't know how the CT Kibra reversed the working memory um, deficit in terms of like which cell type, like as you're mentioning. But this paper from Mount Sinai could give us some clues as to what cells to look at for sure. So this will be great for us to to take a closer look at. So thank you for bringing that to my attention.
Oh, no problem. Thanks a lot. And go Bears. I
Yeah.
know you're not there anymore, but
Oh yeah, alumni. It's cool. Yeah.
Yeah,
All right. So according to my PTR, LT, welcome to the stage. I see you unmuted, but I'm not hearing you. Well, I think that give it a shot.
Yeah, can you hear me now?
Absolutely great.
Thank you so much. Yeah, it's a good talk. I really enjoy it. So, Tara, my question is, you use the model you try to repair, is this hyper accelerated, right? CT, the mouse model, correct?
Yes, that's right.
Okay,
Yeah.
okay. And then also the pathogenic tau is also associated with is it also associated with a hyper phosphorylated tau. So is the CT caper you use, did that also restore or like help the hyperphosphorylated tau mouse model? Yeah.
That's a great question. Yeah, so there's the hypercellated tau model does have this. This might sound confusing. The hypercellated tau mouse model has hyperphosphorylated tau in the brain. um,
Oh,
yeah, so
okay.
but that being said, tau is phosphorylated and acetylated on many different residues. So there's many different kind of types of acetylation and types of phosphorylation. And it's very, very hard to get a good understanding in the field of what, like how the phosphorylation and acetylation of each residue plays a role in the disease in humans. So that's actually a very big challenge in the field. Um, and I think a lot of it could be due to the fact that maybe, there's different stages of the disease. Maybe in different people, there are slightly different, manifestations of this phosphorylation or acetylation. And so that makes it hard to model in mice, right? And so we don't have a model of tau hyperphosphorylation per se, but we do have other mouse models of tauopathy. There is another one that we've been working with that are called PS19 mice. And these PS19 mice are interesting because they have the expression of human tau in the brain. And the human tau has a mutation that causes dementia in people. So there are mutations on the tau protein that cause dementia in people. And so we can model that in mice. And so those PS19 mice also have hyperphosphorylated tau in the brain.
And you tried it, right? Did you?
Yes, yes. And we've done the LTP study in the PS19 mice. This is unpublished data. The LTP was repaired with the CT Kibra in the PS19 mice. And we have done the memory tests, but we don't have the results yet. So this is, one of the models we're interested in. As I said earlier, we're also interested in testing the CT Kibra in the mouse models that also contain amyloid pathology, right? And there are several models that we could use for that. But yeah, so the question is, how broadly will this have an effect across the different kind of disease pathologies? And yeah, so we're very interested in following up on that.
That's great. So you think the CT Kibra is sufficient to restore the this LTP functions?
Yes, in the tau, both in the tau KQ mice and in the PS19 mice, it's sufficient to restore the LTP.
Okay.
So far, that's what we know so far. Yeah.
Thank you. No more questions.
Thank you so much, LT. And yeah, I think there's a comment here or a question at the room chat. So yeah, please go ahead if you have one more question. Mustafa, just type it. I'm going to read one of the questions that was sent to me before I go to your question. So there is a comment from Arnel. He wants to ask what implications do these findings have for the development of personalized treatment approaches tailored to individual patients based on their Kibra and Tau levels.
Yeah, I mean, this is kind of what we want to do. We want to not just try to see if we can make this a therapy, but we also want to figure out who would benefit the most from it. And this is very much kind of a personalized medicine approach. And so that's why we're continuing to try to better understand the biomarker aspect, and also the genetic um, possibilities in terms of whether the polymorphism could give us an indication of um, the susceptibility to the Kibber deficiency that we see in tauopathy. Um, yeah, that's definitely I think very important when considering developing a therapeutic.
Yeah, thank you, Tara. So I'd like to read the question here by Mustafa. And he said his question is, what are the clinical implications of altered levels of Kibra in the human brain and the CSF for early diagnosis and monitoring disease progression in tauopathies?
Oh yeah, I mean that would be our dream, that this monitoring Kibra levels would tell us something about the function in the brain. Um, and that's kind of where our what our data is supporting so far. But for future work, we have to do larger studies. The study in our JCI paper was just kind of the first evidence, and it was a smaller scale study in humans. So I believe we had about 25 subjects for the CSF. So we're hoping to get more than that, to and to run more analyses on the samples, together with the cognitive um, uh, indications, and even to do a longitudinal study would be awesome, for that, to see how Kibber levels change in people over time. So these are all future directions for us to help us get closer to really making a clinical impact for this.
Yeah, thank you. And I think there's another question from Mustafa. What are the underlying molecular mechanisms linking Kibra levels to synaptic dysfunctions observed in tauopathies?
Yeah, so I touched on that just briefly at the end, where we think the deficiency of Kibra, we think, is uh, uh, could contribute to the lack of stability of the PKM zeta, which is a kinase that plays a role in the expression of LTP at the synapse. We also think that we have some relatively new data that's not in the JCI paper, but we also think that the Kibra in general binds to other PKCs, which are other kinases that are similar to PKM zeta, and that could also play a role at the synapse. And so so our data so far is suggesting the mechanism linking or between the Kibber deficiency and the synapse impairment is through this PKM zeta and maybe the PKCs, like all of the PKCs that bind to Kibra, could be affected, their stability at the synapse.
Yeah, thank you. So, how might be the discovery of Gibra's role in synaptic repair impact our approach to treating other neurodegenerative disorders characterized by synaptic dysfunction? So, yeah, maybe this is just like a curiosity question because Kibra is very powerful in doing this. Synaptic repair. So, if there are other neurodegenerative disorders, uh, yeah, maybe have you thought about its possibility, Kibra's possibility to be used in that aside from Alzheimer's?
Oh, yeah. So that's a great question. I think uh, we haven't looked at it, right? However, um, I think there are also many different types of synapses in the brain. I really was just talking about the glutamatergic synapses and in particular the ones in the hippocampus that are important for encoding memory. So it is very focused on this particular type of synapse that's also the most affected in Alzheimer's disease. And so that's why, part of the reason why we're focusing on it. But yeah, we don't know if it could play a role in others neurodegenerative diseases, aside from maybe other tauopathies um, that that we think it would also affect. Um, but yeah, I think that the way that we would need to figure that out is through mouse models, right? There's mouse models of other neurodegenerative diseases. But yeah, we haven't looked at that yet.
Yeah, thank you, Tara. So, yeah, I hope that you will get your lab will be getting more funding so that you could continue your interesting discovery about the Kibra and how it relates to other fields of study and address the interesting questions that we have in science. So, you mentioned a while ago that you might try to look at also because you were just looking at tauopathy, but on the beta amyloid point of view. So, are you going to do a study on the Gibra and vis-a-vis the beta amyloid effect on Alzheimer's in the future?
Oh yeah, yeah, yeah. I think the what we would probably do is use one of the mouse models, like the 3x. There's a 3xTG mice, which have um, amyloid beta pathology, but they also have tau pathology. Um, and so it's one of the models that have both amyloid and tau pathology that many people use for doing preclinical tests um, on therapeutics. So we'll test the peptide therapeutic in that model, for example. Um, I wouldn't, based on what we know, I wouldn't expect the Kibra peptide to have a direct effect on amyloid beta, but I think it's possible that we could still see the same repair mechanism. Um, could still be possible despite the presence of amyloid beta, but of course, we have to test that to see whether it's true or not. Yeah.
Yeah, thank you. So I see that there are questions here, I think, by Mustafa on the room chat. But just one more question before I go. Um, So recently, there has been a deluge of studies in artificial intelligence, particularly when the large language models came in, the generative AI came in. There has been partnerships in biology, and in fact, according to Eric Schmidt and the other investors, they are putting more investment in biotech and also in study of biology, because we now have a tool aside from the other tools that we have, which is artificial intelligence, to have more discoveries or also disruptions that we will be having in biology. So recently, I think that was yesterday or the day before yesterday, there was a feature about Open CRISPR, wherein this AI, which is of course related to LLMs, they had done a study which exhibited 95% editing efficacy across cell types with low off-target rate and is compatible with base editing. So this is just like an example, but that basically what AI does is, of course, it has it gives us a little bit of superpower by prediction. So that's why I was thinking, because you are trying to look across the other pathologies of that might be looked at in terms of correlation with the Kibra, might be a design. Maybe using artificial intelligence be quite helpful to look to use. Of course, the implementation would be in the experiment that you will be using, but it might reduce using LLMs in AI might reduce the time and also the lots of data that you might need to sort out and before you execute the experiment. I would just like to ask, what can you say about this?
Yeah, that's really interesting. Thanks for raising that. So we haven't done this, uh, uh, or we haven't been planning to implement this kind of strategy, but I can absolutely see the advantage of it, and particularly in large scale, dealing with large scale data. And uh, I could see it being maybe also useful in human studies, like with large, it's always good with human studies to have as large cohorts as possible, I would think, because humans in general are quite variable. Um, and so I could see that us, in the work that we're doing on human studies with our collaborators. I could see this also being a potential direction that could be useful. So thanks for that suggestion.
Yeah, thank you so much. I'm very excited for you and your lab. This is really very, very helpful to humans. So we are rooting for the continuation of your study and for your discovery in your paper. So I'd like to pass the mic to Bob to read, I think, Mustafa's a question, or if anyone here on stage will have a question, just flash your mic later on, because we are going to ask Tara later on in about 6 minutes, if how much more time does she have for us. So Bob, would you like to read the question of Bolkar or of Mustafa on the room chat?
Certainly I would. Over what horizon would you identify new potential therapeutic targets for the development of more effective treatments?
Yeah, so this is, I think if I understood the question correctly, this is what everybody wants to know. I think that there's been, certainly in the last decade, there's been a much bigger push um, in our country, to fund research to um, try to come up with more different strategies to treat uh, diseases like Alzheimer's disease, which is such a big problem in our country now. Many people living with Alzheimer's disease, and there's going to be many more moving forward. So, I think that with so many of us scientists working on it, there's more and more ideas for how to design new treatments. In the past, the major focus was on reducing amyloid beta levels. And, we have some therapies now that are showing some promise, at least, although some would argue that maybe it's not quite good enough. But it's definitely something that I think is a good thing. Uh, but I think there's going to be more in the next decade. So I'm very hopeful based on the amount of research that's been going on in the last decade. This takes a while to go from the bench to the bedside, as they say. It takes a number of years because research is hard, and we all try our best, and especially new ideas. So, but I'm very hopeful in the next 10 years, at least, that we'll have more possibilities for therapies.
Well, I thank you, Doctor. I think Volkar has a comment at this point.
Yeah, I was wondering on the humans, the tau accumulation in the brain cause progressed Alzheimer disease. Is there a certain time frame until what extent the tau accumulation creates Alzheimer disease? And is there a way to detect the tau accumulation in the early stages or even earlier than Alzheimer's in the human brain?
Yeah, so there are ways that other researchers are trying to detect tau in humans at early stages. Um, there are methods to do brain imaging with tracers that bind to pathological tau in the brain, and then they can do brain imaging and kind of track where the pathological tau is in the brain. These approaches are not necessarily widely used. They're not. I think there's they're being used for research purposes, and then hopefully someday they'll be clinical someday soon. Uh, the other way that is being developed to look at tau in early stages is tau levels in either cerebral spinal fluid, which I showed some of our data, or tau in serum from the blood. And so people are definitely making efforts to try to find ways to basically detect Alzheimer's disease before it starts. Because that obviously would be the best case scenario so that you could treat people before the disease gets bad. So I think this research is making progress, and yeah, so hopefully in the near future that this will become part of clinical practice. But right now, I think most of this is really still at the research stages.
But if we, for example, for the sake of argument, if we detect tau accumulation in the brain in the early stage, with our current technology and current knowledge, is there a way to stop the progression, or is it just to slow down the progression?
Yeah, so the way people are doing that is they're trying to knock down the expression of tau, like reduce tau levels. Or using antibodies to reduce tau levels, which is the approaches they've used for amyloid beta, or similar to the approaches for amyloid beta. And so, yeah, reducing the tau protein if you reduce it after kind of disease onset, then you would slow, I would expect that it would slow the progression. But if there was a way to reduce it before the accumulation, then it could be protective, maybe. Protect the brain from having any accumulation of tau. I mean, that would be amazing. But right now, I don't think we have that, as
But
far as I can tell.
so basically speaking, if we find a way, theoretically speaking, if we find a way to detect tau accumulation in the first place, and with our current knowledge, it might be possible. And if it's not progressed to Alzheimer's, you could have stopped it in the first place with our current knowledge and technology. Correct?
Um, I, I, I think that's going to be much, much harder to do. And yeah, so theoretically, maybe. But I think based on current technology, I'm not sure that that's going to be possible in the short term. You know, but who knows? I mean, technology is advancing every day, so I also, don't want to, I want to be hopeful at least, but also realistic that slowing the disease progression, I think, is also very good. That's better than what we've had, up until now, right? And that, positive steps are good steps, I think, especially for, those who are suffering, right? So, um, yeah.
But treatment is one thing, but prevention is another thing. So on the prevention side, if you find, so basically speaking, if you find a way to detect the tau accumulation, it will ease down the future Alzheimer cases. But treatment on the current case is a different subject, I guess. Those are both different subjects.
Yes, 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 absolutely.
Thank you, Bokar. I want to touch LT. Are you ready?
Yes, I'm ready. Thank you. And Tara, so speaking of early detection, so I look at the slides 19. So I have the question that when you talk about the early detection, it's practical just to look at the CSF level of a pathological tau in the CSF and also the serum you mentioned. But on the slide 19, it's funny because I just realized you said less kibble in the CSF and more kibble in the brain is correlated with good cognition, and you have less of the pathological tau. So how did the kibble really normal function? Is that the difference in the CSF versus in the brain? I thought that more kibble may be good for the cognition because it repairs the LTP, right? The function.
Right. So you're asking,
Yeah. The
in healthy people, is that
yes.
in
Yes.
healthy people? Yeah, in healthy people, we think there's more kibber in brain and less kibber in CSF.
Yeah, so
You
as
know,
if there's a check, right? As if you have as if what happens, you have more cable in the CSF with that, then you become like a cognitively impaired.
yeah,
So
so
that
that's the thinking that, the idea is that if uh, if it's possible, to test people based on how much kibber they have in their CSF to be able to know, how susceptible they would be to memory loss, for example. And so if you'd have more kibber in your CSF, that might be a bad sign. That's what our data is suggesting. Um,
then go ahead.
no, that's it.
Okay. Okay. So then just a quick follow-up is that Because you mentioned kibble is like a bands, like about 20 different like proteins in the neurons, right? In those neurons.
Yeah.
So then you probably have a panel screening. So is CSF binding slightly different subset of those 20 among of versus in the brain? I'm just wondering.
I see. I see. Yeah. So there have been studies in the literature done by others showing that Kibra binds to 20 different proteins. It's actually there's quite a few different studies on this on just looking at Kibra interacting with different proteins. So. We have not looked at whether the Kibra in the CSF is binding to anything. We haven't looked at that. And we don't necessarily, so cerebral spinal fluid is kind of, it could be considered that maybe this Kibra isn't really functional anymore, right? Because there are no neurons in the CSF, right? So we would think that the Kibra inside the neurons is the functional, is going to be functional at the synapses, but then the Kibra that's outside in the CSF is probably not functional anymore to some degree. So, we haven't looked at it. So I'm not sure if it's binding to anything or not.
Okay, okay, because in the patient you have more kibble, so that means whatever so that as if there's a leakage, so you have less,
Right.
the brain neuron could not keep all those kibbles in the brain, so they just goes to the CSF. That's what
That's
I think.
right.
Okay.
Exactly. Yeah.
Oh, I have a question.
Thank you. Yeah.
Oh, if I can jump in, I guess. Yeah, just you mentioned like a decade ago about the genes that have been identified with Alzheimer's. And forgive me if you've already mentioned some of this, but made me think of Ronald Reagan. The during or after he had Alzheimer's famously. So the NIH obviously funds this a lot, and so it made me think of in the last decades we had like 70 genetic regions now are associated with Alzheimer's, like the APO is variant. Is a significant factor for Alzheimer's, and also it can cause brain cells to build up abnormal amounts of lipids instead of using them to insulate nerve fibers. And so I was wondering if any of this correlates with that. And also, you mentioned protective variants, so it made me think of APO 3CH, I think, channel variant is a supposedly protective one as well. So, and also the APO4 protein in neurons trigger an immune pathway that leads to tau tangles and cell death. So it made me think of some of that. If you have anything to say about that, if you haven't already too much, hopefully not.
Yeah, that's great that you're keeping up on that research. Yes, ApoE4 is one of the most common and biggest or greatest risk factor for Alzheimer's disease. So if you have an ApoE4 allele, you're at higher risk of Alzheimer's disease. It doesn't mean that you'll definitely get it, but it just makes you higher risk. So it's a very interesting gene. Many people studying it. There is a link between ApoE and Kibra that's interesting. There's a genetic relationship. So the polymorphism that I mentioned for the Kibra gene, there was a paper, a human study that was published by another group that showed that people that had the protective, I think they showed the people who had the Kibra allele that was protective had were protected against the ApoE4 related risk. So, these risk factors, there are many of them now that we are aware of. But the interesting part is that, depending on which risk factors you have, and their interactions with each other, you may be at less or more risk of susceptibility, depending on which ones you have. And they probably, many of them probably interact with each other. So I think there are a number of, human studies that are trying to, better understand the links between them. But there is the one that I know of is there is some interaction between ApoE4 and the Kibra polymorphism.
Oh, interesting. Thanks a lot.
Hi, guys.
Thank you, Kevin. So, yeah, I think the last person who will ask a question is Gu Racket, whom we would like to welcome on stage. Gu?
Hello, everyone. Hi, Tara. Great work yet again in this room. My father died of Alzheimer's a while back, but whilst I was in the care home, I did a sort of unofficial study, and it was more dementia patients. But there was definitely a link between gum disease, gingivitis, um, and Alzheimer's, and dementia patients. It was everybody. That was the only link I could find. They all have false teeth, and there was some work done a while back. I've just if you make me a mod, I might be able to post it the link. But there was some work done on it, and there was definitely a link there, and it was maybe you could interrupt the process, the development of the disease from that. Is it something you were aware of, Tara?
That's really interesting. I uh, I have not heard as much about it, but it
Right.
almost there's also anecdotally, from my own experiences too, you know, um, things like injury or things that can cause inflammation can also be triggers, I think. And there's maybe some evidence in the literature for this that, the brain is not in isolation in the body, right? Like other things in the body could affect the brain.
Mm-hmm.
Um, and so especially if there's some injury or inflammation, which I assume would be happening in gum disease, there's probably a lot of inflammation related with that.
Well, I think it
Uh,
was to do with proteins and things like that. I'm not a neuroscientist, and I don't know how they all work,
yeah.
but obviously, there was definitely a really definite link. And
Uh,
then I was in a room probably a couple of years ago. And it was about trying to put people into deep sleep, and how the effects of this, is it torpor it's called or something, and they were using ultrasound to do it, and they were finding one of the effects was it was actually it stopped Alzheimer's in its tracks. Tara, if you DM me, I'll send you that presentation. I've got it. I've saved it all. Anyone else wants a copy of that as well? It's amazing. It was a Chinese guy, and he was using it to try. Like say they go to Mars and things. How we could put people into a deep sleep using ultrasound, and one of the net effects was some pretty positive things with regards to dementia and Alzheimer's. Anyway, that's all my knowledge gone, and I'll shut up now. But thank you, everyone.
Thank you. Yeah, interesting. Yeah.
Yeah, thank you, Gu. And now we are going to wrap up because we've been here for 1 hour and 45 minutes. So we would like to thank all those people who came on stage and who asked.
Are you muted, Cecile?
Five.
Cecile, you're still muted. We can't hear you. She'll be back.
Can you hear me now?
Yes, we can. Welcome back.
Yeah, thank you. So we are now at the top of the hour, and we would like to thank all those who participated here, Bulkar and Mustafa, and also LT a while ago. And yeah, Kevin, of course. And I take this opportunity to thank our guest speaker, Dr. Tara Tracy, who shared with us their very important research. And we hope that we can invite her again here in Quantum Photonics, because I'm sure that her laboratory, the Tracy Lab, has a lot of very interesting and very relevant research. And you see, Dr. Tracy, that many of our members here in Quantum Photonics are very interested to listen to your future presentations. So thank you. And I'd like to give you the mic if you have some last comments before we close the room.
Oh, thank you all. Great questions, and I really appreciate all the ideas that you all shared, and it's been fun. So thank you for coming.
Yeah. So, all right, we have Darcel here. I cannot mod you.
Cecile, were you still talking? Or I think she was handing off to you, Bob. Is that correct?
I, oh, you welcome back, Cecile.
Sorry, I don't know what's happening. Anyway, I'd like to, yeah, say that before handing the mic to you, Bob, I would like to tell everyone that on the morning after tomorrow, we are going to have a very interesting room with a guest from Aalto University in Europe. And the popular title of the room is Measuring Qubits Evading the Heisenberg Uncertainty Principle, and with a very interesting paper. It's called Single Shot Readout of a Superconducting Qubit Using a Thermal Detector. So please check out our
What
platform.
time is it? Eastern time? What time on Saturday?
It's gonna be at 8 a.m. Sorry, for Pacific, and 11 a.m. for the East Coast. So yeah, just check out the platform that we have. Will be here with Dr. Mutanen. So I'd like to give the mic to Bob now, if he has something to say before giving it to Darcel to help us close the room. Yes, so now, Darcel, let's close the room. And thank you again to our guest speaker, Dr. Tara Tracy. So, Darcel, mic to you.
Thank you, Cecile, and thank you, Doctor, for a great talk. To close the room, we're going to start our countdown from lucky number 6. Is everybody ready?
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 Route
Ready.
6.
Ready.
Route 66.
6.
6.
Kevin 5.
Fiverr.
Barker 4.
Four dirt.
Bob 3.
3 it is.
Cecile Dos Equis.
Dos equis, please.
Good doctor, you're numero uno. Call out our last number, please.
One.
Thank you so much,
Thank
Dr.
you.
See you back in Quantum Photonics soon, and good luck to you and your team. And yeah, see you in the next rooms, everyone.
Bye.
Bye.